Thank you very much. Happy New Year, everyone, and thank you very much to the organizer for this opportunity. I'm a little nervous and I'm really privileged and excited. So I will talk about microbial electrophysiology. That's something I've been trying to work on for a while now. Before I start, I will talk about the company we started a while ago. So it need declare conflict of interest, and we'll talk about a bit of that, what we started, why we started the company, and what we're doing with the company. Although I'm at heart, I'm just basic scientist, and I never really imagined starting a company. And whole drive to me was sort of trying to understand what is life and how life works. Is there a process or matter? That sort of thing is what that fascinates me. And when we think about life, there are many interesting things, but I think one really fascinating thing is that incredible diversity. Some are like a tiny bacteria, some are beautiful and flies, some swim, some like a much larger than us, or some our size. So it spans like a very different time scale and spatial scale. And somehow there is something we can recognize as life. And I think that's really incredible, and that's something really fascinating. But yet, I think one of the heroes of many of us probably is Jack Mono, and he once famously said, "What is true for E. coli is true for elephant." And I can prove he's wrong. It's easy to prove he's wrong. There are many features that is only true in E. coli and not true in elephant. But this quote is really insightful. And as we know, because he was making that point in the context of molecular biology and how genes are regulated. And indeed, there is something common from bacteria to elephant, despite all those diversity, something common in terms of how genes are regulated and controlled. Now, but I think it is also getting clear these genes and sort of molecular kind of thing is not enough to understand biology. And there's something missing. And one concept I think emerging is the idea of that all biological cells and molecules are electric. And particular electric aspect I want to talk about and I've been studying is the membrane potential. This is a electrical potential difference across the membrane. And it's a central topic in neuroscience, but outside of neuroscience, not many people really care about this membrane potential dynamics. But yet this is something exists in pretty much all cells. There are a few arches which has the opposite membrane potential, but usually inside is negative and compared to outside. And usually this is sort of regarded as a background homeostatic thing in biology, but there are evidence suggesting this is actually dynamic. And so this was a project I was involved during my postdoc time in Grosswell's lab in UCSD. This is bacteria biofilm, and where we are looking at was membrane potential. And their bacteria in biofilm coordinate their behaviors through changing membrane potential, and that ended up also linking to the metabolic coordination. And then there's a growth oscillation associated with this electrical communication. And that was the example we came across, but in literature, there are many examples that electrical potential and electrical polarity seems to exist, and then seems to be doing something in like wound healing, tissue regeneration, development. Cancer cells have a different membrane potential than the non-cancer cells. So there is something there. And with that, I think there is an emerging idea that maybe electricity could be a sort of paradigm that we can generalize to try to understand the different biological phenomena. And perhaps also we can start thinking about sort of general ways to use electronics or electric stimulation as a ways to control or engineer cells. So, and this is complementary to genetic engineering, in my view, rather than sort of competing. But there are things that this type of electric physiology-based control may be more useful. So my research group focuses on bacteria. So we're interested in sort of trying to develop tools to control bacterial membrane potential, or at least outer membrane potential, as well as try to understand to what degree bacteria are using this electrical signaling. What we know is in bacteria, there are processes like ATP synthase or motility, membrane transport. They are driven by membrane potential, in part ion multiforce. And there are examples showing antibiotic resistance and tolerance are linked to that. But still, that dynamics is not really understood. So that's where we're really interested in. And central to all cells, again, is a link to metabolism. So this electrical potential gradient across the membrane is tied to central metabolism. I think most known example is ATP synthase that is driven by proton multiforce, and that's pH gradient and then membrane potential. But in bacteria, there isn't much of the pH gradient, so the membrane potential is a major driving force of the ATP synthase. And so there is something, a fundamental sort of livingness, if you call, that is linked to this membrane potential. And this neural signaling is also in many ways actually linked to the neurons metabolism, like CO2. And there are many neurotransmitters that's actually one or two pathways away from central metabolism. So I think there is some like a fundamental link between them. And with that mind, sort of kind of question we were wondering was, if we give electric stimuli, like a short pulse of electric stimuli, what kind of change can we cause to membrane potential? And James, postdoc at that time in my group, and the corner student at that time were working together. We built like a simple device, gold electrode, and coated dish. And you can see some agri glue, not really professional, but we're really happy with that shiny gold dish. And we can measure membrane potential of bacteria using fluorescent dye. And we can also see individual cells. And our experiment is simple. We have that device. We can see individual bacteria. We just measure membrane potential. And what we are expecting was, we know those bacteria have ion channels, and many ion channels are voltage sensitive. And if we ignore all shapes and then complicated biochemistry of bacteria cells and then think that as a sphere, we can use like a simple equation to sort of, infer what might happen. And that's proportional to the external electric field and the angle and then radius. We can cause change to membrane potential. And if that change is large enough to open channel would open. And if there is a large enough chemical concentration gradient, we would have efflux of potassium. And that's sort of what we observed. So in this experiment, when you see a lightning sign, that's when we apply AC field, 2.5 seconds. Whole movie is 1 minute, or 30 seconds, sorry. And so immediately after we apply electric stimuli, they all more cells lights up. But one thing we noticed was not all cells lights up, like this cell lights off, that also lights off. There are some cells lights off. And we thought, well, maybe it's linked to cell vitality because membrane potential is so tightly linked to the cell state. To test that idea, what we did was we give UV light to that center region and then stress cells. So you give 30-second UV and then observe growth for 1 hour. If we zoom in, we can see some growth here, but no growth here in the center region. Yeah, so some growth here, but no growth here. If we just look at those cells, we cannot distinguish anything. They just look identical. And then we apply electric field across 2 electrodes. And as we expected, the cells UV treated, we see a signal going down instead of going up. And we wondered why this identical stimuli cause opposite direction response. We borrow a mathematical framework from neuroscience. It's a phenomenological model, not really grounded on biophysics, but phenomenologically, we can recapturate many of neural membrane potential activities. So we thought that's a good starting point. And I'm gonna skip this part a bit, but if anyone is interested, I'm more than happy to discuss and then explain. But we were able to recapturate that opposite direction response using that model by changing one parameter. That's 10 and the 0.1. Apologize for that. So the message we got, or the interpretation we got from that simulation is how quickly membrane potential change basically affect the direction of change. And from that, how we interpret that data in the biological context was when cells are really healthy, they accumulate lots of potassium by using a lot of energy. So they use a lot of energy to accumulate potassium. So when electric shock opened the ion channel, there's enough chemical potential gradient to drive efflux of potassium. So we can see the membrane get hyperpolarized, going further negative. But when cells are UV inhibited, they cannot maintain this very energetically expensive process. And when the channel opens, there's not much to happen. So our current understanding and interpretation is that electrical stimuli cause depolarization to the membrane. We don't still understand exactly how that works. This is something we're really interested, but I haven't managed to secure enough funding and resources to dive into in depth. But yeah, we don't know exactly how that channel opens and how that depolarization happens. It may be directly by the electric field or some electrochemical reaction indirectly cause that. But the point here is basically depending on the the energetical state of cells, which mainly used to accumulate potassium, we will get the different responses when ion channel opens. And if that is true, it doesn't really matter how cells are inhibited, because at the end of the day, if cells are not doing well, their energy maintenance will be collapse. So we tested antibiotics, ethanol, CCCP. They all target different parts of cells' function, and they cause trouble to cells' function. And in all cases, when cells are stressed, the signal just go down instead of going up. So in sort of nutshell, we get basically two different responses depending on the cell's state. So that input here is electrical stimuli, and the change in membrane potential, we have either going up or down. And we tested that with different bacteria. And so original study, we used Bacillus subtilis. That's our main model organism. But then we tested E. coli, Staphylococcus, and the Lactobacilli, and they all seem to show that opposite direction response. So we thought maybe we can extend this pattern to see if this pattern hold in eukaryotic system. And there is no obvious reason to believe that might be the case, especially because eukaryotes have mitochondria where ATP synthesis happens. So that in prokaryotes, everything happens on the membrane that facing to the outside world. But in eukaryotes, the metabolism is separated and insulated from outside world. So when electric field is applied, most likely there won't be any like direct change to mitochondria. And to test that idea, we haven't really tested like mammalian cells yet, but as a sort of starting point, we used yeast, Saccharomyces cerevisiae, as a model organism. And our experiment is, again, very simple. We have that electrical stimulation device, and we just measure how membrane potential indicator change over time after electrical shock. And Thalisa, in my group, did that experiment, and she used different markers for membrane potential, plasma membrane, and the mitochondria membrane potential. But what she found is the plasma membrane potential changes, and that change corresponds to cellular stress. So this is similar to bacteria experiment. We apply UV to that center region, and then apply electric shock afterwards. And as you can see, that the UV-treated region and the non-treated region respond differently. Now, one big difference we noticed with yeast is this happens over a course of 20 minutes to 30 minutes, much slower than E. coli or Bacillus. So the E. coli response is a bit slower than Bacillus, and yeast response is much, much slower. So there is some differences. But in terms of how they respond and then how that relate to the cell's state, there's something conserved, seems to be conserved. So we thought maybe this could be useful also to measure cells sort of vitality, cell vitality, or some sort of cell livingness. So we're now proposing BEAST, bioelectrical antimicrobial susceptibility testing, to test the sort of key concept. This is still yet to be established. It's just idea at this point. But as a sort of starting point, what we did is we measure growth rate in liquid culture using different antimicrobial compounds, and then work out the growth rate. And we, on the other hand, we also treat cells with different antibiotics, antimicrobial compounds, and then did electrical stimulation and just measure intensity change before and then after 20 minutes. And then we compare them and then map them out. And we found reasonable correlation between them. It's not perfect, and we still have some uncertainty. Like here, we have quite large variations, which we don't know exactly what originate, but there is some promising result showing there's a nice correlation. Now, key difference between this assay and that assay is this assay can be done within 1 hour. This assay takes overnight. So potentially we can get same information much faster. And the other advantage of that is potentially we can get individual cell data. We still don't know if at the individual cell level, if we can correlate the growth rate to response yet. But this gives us the idea and the potential that this could be useful to measure cells vitality at the single cell level. Now, this could also be useful in our society because bacteria or microbes are everywhere. Some microbes cause infections or contaminations. And there are many places, or environmental monitoring, soil health. There are many places we need to measure live bacteria. But the most common technique people still using is plating. This method is really robust and useful for many microbes, but it's slow. It takes overnight. And for some microbes, you cannot culture, or it takes weeks to culture cells. So that's where we started the company, and we thought this could be potentially useful for the society. And yeah, last year we got some business award that was quite exciting. And still, company is still early phase, not really making any profit. But yeah, it is an exciting time. And one of area with company we're now exploring is in the context of antimicrobial resistance. This is becoming a more and more big issue. Normal infection may become uncurable when AMR becomes so common. And one area we think this technique could be useful is in the blood infection, because that rapidly progress. Right now, when patient was detected with blood infection, physician gave general antibiotics and then send the sample to laboratory. And then in laboratory, they do test to make sure that antibiotics works. And then if the infection happened to be a resistant one, then on day 3, they know which antibiotics works best, and then they can change the course of action. If the fast antibiotics works, that's fine, no problem. But this is becoming more and more problem because there are more and more resistant strains. What this sort of rapid test could do is it can give result on day 1, and that means that better patient outcome because the optimal treatment is done earlier, and also shorter bed time, reduce healthcare cost, and that also means we don't need to use unnecessary use of antibiotics, which is known to was the problem of the AMR, emergence of AMR. So we got a large grant last year from NIHR, that's a UK NHS-linked funder, and we're now testing that possibility in collaboration with me as a Warwick University academic, and then also Leicester, University of Leicester, and the NHS hospital. And with Warwick colleagues, we are now also making socioeconomic model. How much impact can it make? And also we're keen on making patient public involvement using with Cheetah. And yeah, we're getting more data, improved data analysis, developing graphic user interface. It's very early phase, but it's quite exciting development. That's almost all I have to say. but I want to use this opportunity to advertise. I was recently really fortunate to get a grant from Liver Hume Trust. This is more fundamental science-based project. We are really interested in understanding how this electrical communication happens in cells, as well as yeah, get the what does that mean, and then to cell physiology, and also what sort of information can they code. So I'm recruiting a postdoc. Yes, the deadlines this month. So if there is anyone maybe interested, or you know who may be interested, please encourage them to get in touch with me. There are also PhD studentship. So yes, I'm really advertising, and as advertising, I'm organizing, convening a session in ASM microbes in June in LA, called Energizing Microbial Cells. The abstract call is open now, so if there is anyone who may be interested, please consider submitting abstract, or please come by to that session on June 22nd. With that, I would really like to thank everyone. I was really fortunate to work together in my group, as well as all collaborators. For the stories I shared today, I want to make a big shout out for James Corner, Talise. But yeah, everyone did. Yeah, I was really lucky to get the right people, and also funding. And yeah, thank you very much for your attention. And happy to take questions if it's any. Thank you.